So in this video 4 of our course G05 on antibody analysis, we're going to talk about IG sequence annotation here. So firstly, let's consider that there's a wide diversity of databases associated with antibody diversity. Chief among them would be OAS, the observed antibody space, which contains billions of FE sequences from 68 BCR-seq studies containing over 600 million sequences. There's a website here that's still accessible. Another one is AbDiver, which contains the curated B-cell receptor omics data and mutations for different antibodies and their design and engineering. Another one is Rapid Repertoire Sequencing Dataset Analysis Platform with an integrated antibody database, which contains human heavy chain repertoire analysis. And there's more beyond this. This is just 3 key ones. There's more listed at this paper at this site. And therefore they contain there's a lot of different options that are present. So we're not going to go through all these in this course, but just to note, there's a huge diversity of ones, chiefly centered around humans, obviously in SARS-CoV-2 and so on. But then if looking at those for livestock, we have to think a bit further. So we know that there's great diversity for IG alleles, particularly at the FR1234 and CDR123 regions. And we know that annotations are often very human or mouse centered. And therefore, if we look at rarer livestock species, the antibody structures may differ there. An example of this is the CDR3 region in cattle is much longer, and therefore it needs a slightly different type of modeling. So the tool we're going to look at uses is based in the NAR-CI database, which uses the numering schemes of IMGT, Chotia, Kabat, Martin, or AHO. And these kind of contain, this reflects a diversity of antibody and TCR alleles that we know exist. And the tool we're going to use, IgMAT, mainly is based on IMGT data, which I misspelled here. So what we're going to use is this tool, immunoglobulin multi-species annotation tool, which is a simple tool for annotating segments of VDJ protein sequence. It works very well for less common livestock species. It aligns the input given, which is a FASTA style protein sequences or a set of protein sequences, to the IgMT database using different number schemes using hidden markup models. And extend this to these other diverse livestock using created germline antibody sequences. And notably, it tolerates longer CDR3s, for example, by examining the extent to which there's multiple matches from different hidden Markov models. So those of you who are maybe unaware of hidden Markov models, they are models of Markovian processes. What we're essentially interested in is a sequence of events where the probability of a previous state I depends only on the sequence of events where state I depends only on the previous state. So we can infer state I based on the previous state. However, it's tricky. Inferring this state X is tricky because we can only observe YI, which is associated with X, and its states are influenced by X, XI. So we're learning about X via Y. And we should note that I minus 1 is independent of XI, and likewise for Y. So Y is proxy for X. So we can think of an example of this, such as binary classification tools that are looking at a series of ones and zeros, where we have two states, 1 and 0. And we think of these about their emissions and transitions. Transitions are switches from one state to another state, like 0 to 1 or 1 to 0. And emissions are the observations of the states themselves, or the probabilities of observing that particular state. And we can extend it as well, that when we're thinking about multi-sequence alignment containing protein sequences and nucleotide sequence, we define number of states. For nucleotides, that's relatively easy. You have 4 or 5 states. for protein sequences, it's more complex because there's typically 20 states. Of course, there are certain proteins that are certain bases, there's nucleotides as well that are organism specific, but there's proteins as well that are whose usage rates differ across organisms, and therefore not all amino acids may be included for certain organisms. So chief among the antibodies of interest, organisms of interest here are horse, mouse, camel, human, pig, chicken, and there's more information at this website. So what we're essentially looking at is these V and J sequences of interest. And that's where we're going to focus in the demonstration video on this area. We'll look at how we can use this tool to annotate immunoglobulin sequences that are of interest where we may not know precisely which segment corresponds to which region of interest.